you ever wondered what makes bacteria different from your cells? Today we're diving into the structure of a cell and uncovering the roles of its fascinating organelles. From simple cells with no nucleus to complex cells with organelles, we'll explore how life is organized, one cell at a time. Welcome to CM Explain Science Hub. Don't forget to like, subscribe and hit that bell icon for more science videos. A cell is a basic structural and functional unit of life. All living organisms are made up of cells, and they are either unicellular, that is made up of one cell, like in terms of bacteria, or multicellular, made up of many cells, for example, humans, animals, and plants. Cells are in two types. We have prokaryotics and eukaryotic cells. Prokaryotic cells, these are simple, small cells that lack a nucleus and membrane-bound organelles. So, what makes a cell prokaryote? Well, first, they don't have a nucleus. Instead, their genetic material, the DNA, just floats around the cytoplasm inside the region called the nucleoid. They have the capsule, they have a cell membrane, a cell wall, plasmids, ribosomes, pili, and the flagellum. The nucleoid. This is the region inside the prokaryotic cell where the cell's genetic material or DNA is located. Key points we need to know about the nucleoid: it has no membrane. Unlike in the nucleus of eukaryotic cells, the nucleoid is enclosed in a membrane. It's not enclosed in a membrane. It contains a circular DNA known as plasmids. Plasmids are small circular double-stranded DNA molecules found outside the main chromosomal DNA in prokaryotic cells like bacteria. They are extra chromosomal, which means they're not part of the main DNA nucleoid, but they carry useful genes that benefit the cell. Key features we need to take note of: plasmids. They are small and circular, typically contain a few thousand base pairs, smaller compared to the chromosomal DNA. Another point is independent replication. Plasmids can replicate on their own separately from the main bacterial chromosome. They carry special genes. They often contain genes for antibiotic resistance, toxin production, enzyme production, virulence factors, which these are factors that increase disease causing ability. Plasmids can be transferred between bacteria through a process called conjugation. Plasmids can move from one bacteria to another via the pili. This helps spread useful traits rapidly, which is good for the bacteria but not good for the host. DNA is usually one circular chromosome. In prokaryotic cells, the cell membrane, also called the plasma membrane, plays a vital role in survival, structure, and function of the cell. Key features of the cell membrane: it is made up of a phospholipid bilayer, just like in eukaryotic cells. It has no cholesterol. That is one difference we need to take note. In eukaryotes, we have cholesterol. In prokaryotes, we do not have cholesterol. It is located beneath the cell wall. The membrane lies just inside the rigid cell wall made up of peptidoglycan in bacteria. It has embedded proteins, which are proteins which help with transportation of substance, cell recognition, energy production. The cell membrane is the site of important processes, for example, respiration. Since prokaryotes lack membrane-bound organelles like the mitochondria, the job of respiration happens here in the cell membrane, and also photosynthesis in photosynthetic bacteria. It also happens in the cell membrane. As any other cell membrane, it is selectively permeable as it regulates what enters and what exits the cell. Mesosomes are folded regions of the plasma membrane found in some prokaryotic cells, especially bacteria. They appear as pouch-like enfoldings of the cell membrane under an electron microscope. Some key features of the mesosome are they look like finger-like folds or vesicle-like extensions of the cell membrane. The functions, it is previously thought to say they help in the cell wall formation during division of the cell. It is also believed to say they increase the surface area, hence helping in the respiration and photosynthesis for some photosynthetic um, prokaryotic cells, but it is not clearly established to say these are the functions. The bacterial cell wall is a rigid outer layer that surrounds the cell membrane and gives the bacterium its shape, strength, and protection. It is one of the most important structures in a bacterial cell. The functions are it provides structural support. So it helps the cell from cell lysis, that is bursting of the cell due to osmotic pressure, that is from osmotic gain. It protects the cell. It acts as a physical barrier against harmful substances and environmental stress. It maintains the shape of the bacteria. The shapes, for example, a bacteria or a prokaryotic cell called cocci has a spherical shape. Bacilli has a rod-like shape. Uh, Spirili, yeah, it has a spiral shape. Yeah, okay. It is the target for antibiotics. Antibiotics like penicillin attacks the bacterial cell wall by preventing its proper formation. The main components of the bacterial cell wall is made up of peptidoglycan, which is basically proteins and carbohydrates. One thing that we need to take note when we talk about peptidoglycan is gram staining. So it can either be a positive or a negative. Gram positive bacteria stains purple, while the negative one stains pink or red. A capsule is a thick, sticky layer that surrounds the cell wall of some prokaryotic cells, especially certain bacteria. It is not present in all bacteria, but when it is present, it serves important roles in protection and survival of the cell. The key features of a capsule is that it is mostly made up of polysaccharides and sometimes made up of polypeptides, or it can be a mixture of both polysaccharides and polypeptides. It is a protective layer that shields bacterium from desiccation, and that is drying out. It protects also the bacteria or the prokaryotic cell against phagocytosis by white blood cells. It also helps in attachment of the bacteria or prokaryotic cell to the host tissue or surfaces. This is important in infection. Also, the virulence factor capsules make bacteria more dangerous by helping them evade the immune system. Proteins, the synthesis mechanism of the cell. Prokaryotic ribosomes are 70S in size, made up of two parts, namely the 30S small subunit, and a 50S large subunit. They are made up of both RNA, to be specific rRNA or ribosomal RNA, and protein, giving them a complex but efficient design. The functions of these 70S ribosomes is that they read the mRNA that and join amino acids together to form proteins. The 30S subunit decodes the genetic message, while the 50S subunit forms peptide bonds together. Together, they carry out protein synthesis, and that is translation, of course. The importance: without ribosomes, no proteins will be formed, and without proteins, no life. In medicine, many antibiotics target the bacterial 70S ribosomes, stopping their protein production without harming our large 80S ribosomes. The pili and the flagella. So these hair-like structures found on the surface of the bacteria or the prokaryotic cell. It can be used for attachment or movement. Now, note that not all prokaryotic cells have these features. Only certain types or specific prokaryotes have these. The pili, singular, known as the pilus. So these are short hair-like projections on the surface, shorter and thinner than the flagella. They're made up of a helical structure protein called the pilins. So the functions: it is used for attachment. The attachment of the bacteria to the host cells, tissues, various surfaces, which is important for colonization leading to infection. They are also important for conjugation, which is done by a specialized pili called the 6 pili or the F pili, which facilitates the transfer of plasmids from one bacterium to another in this so-called process conjugation. They also act as environmental sensors, helping the bacteria to respond to stimuli in the environment. When we talked about phagocytosis, these pili, they act as anti-phagocytosis, which helps the bacteria resist the phagocyte engulfing. The flagella. This is a long, thickened, whip-like structure made up of protein known as flagellin. It is also a helical protein. It is used for locomotion, and it has about 100 revolutions per second. The functions: it is used in mobility, locomotion, or movement of the cell. It rotates like a propeller, allowing the cell to swim in liquid environments. It is also useful for chemotaxis, which is the bacteria is able to swim towards nutrients and away from harmful substances, for example, antitoxins. A recap on the prokaryotic cell. Thanks for watching. Don't forget to like, subscribe, and share the video. Remember, science makes the invisible understandable.